In this section, we are going to go over some important background information of things that you will need to know to really get a grasp of the neurophysiology. First, let's talk about 4 types of ion channels that you can find in neurons and which will be covered in several of the activities of this PhysioEx. The first type of channel is a leak channel, and as the name suggests, these channels are always open, meaning that ions can always leak through them. Ions will move down their electrochemical gradient, and so we'll talk about specific ions and electrochemical gradients a little bit later on. The second type of channel is a ligand-gated ion channel, also sometimes called a chemically gated channel. These channels have a gate, meaning that ions are not able to freely move through these channels unless the gate is open, and the gate is only opened when a specific molecule binds to the channel. For example, neurotransmitters are molecules that typically bind to ligand-gated channels on the postsynaptic cell that a neuron is talking to. The third type of channel is a voltage-gated ion channel. As the name suggests, these are also gated, meaning that ions can only pass through them if the gate is open, not when the gate is closed. Voltage-gated channels open and close based on changes to the membrane potential. And we're going to talk a little bit more about membrane potential later on. There are also mechanically gated ion channels. These are ion channels that again have a gate which controls the ion movement, but these channels, the gate is opened or closed based on movements of the plasma membrane. So if the plasma membrane of a cell is distorted, say for example like when you touch your skin, that distorts the plasma membrane of the skin cells underneath the area that you touch, and that would open up mechanically gated ion channels. Now we will review the basic structure of a neuron. So a neuron can be divided into 3 main regions. First, we have the receiving region. This is composed of the dendrites and the soma, or cell body. And the receiving region is the area that is going to either be stimulated directly by some type of stimulus, and this is where you will get graded potentials, also known as receptor potentials, which we'll cover in a later activity. So this is where you will find ligand-gated channels and mechanically gated channels, would be in your dendrites and your soma. The difference between the dendrites and soma is that the dendrites are just there for receiving messages from different types of stimuli, whereas the soma can receive messages and also carry on normal cell functions. This is where you'll have your mitochondria, your Golgi complex, your endoplasmic reticulum, your ribosomes, etc. The second region of a neuron is the conducting region, and this is made up of the axon hillock and the axon. So the axon hillock is the area where the summation of all of the different graded potentials from the receiving region are added together or summated, and the neuron decides whether or not it's going to fire an action potential. So a quick way to summarize the function of the axon hillock is to say that this is the region where action potentials are generated. The axon is a long, slender process that is going to propagate the action potential, which means the action potential will travel away from the cell body or soma along the axon. And neurons may or may not have myelin covering the axon. If myelin is present, then it provides insulation and allows the signal to travel faster. The conducting region of a neuron is where you will find the voltage-gated ion channels. The third region of a neuron is the output region, and this contains the synaptic terminals, which are also called axon terminals. This is where you will have the synaptic transmission, where you will have the little vesicles in the synaptic terminals that will release neurotransmitter into the synaptic cleft to talk to the next cell, either a neuron or some other cell in the body. Now we will discuss membrane potential and related terminology. So membrane potential can be thought of on the simplest level as a measurement of the difference of electrical charges on either side of a membrane. So if the charges had no difference, so the total number of positive and negative charges on one side of the membrane equaled the total number of positive and negative charges on the other side of the membrane, you would have 0 millivolts potential, meaning that there would be no difference in charges. So I've shown that here on our little scale. But that is not the case in cells in the human body. All human cells have more negative charges on the inside and more positive charges on the outside. So in a normal human cell, there is a difference in electrical charges. So you can think of the cell as being polarized. In other words, think of it polarized, meaning that in one area it's got more negative charges, one area it's got more positive charges, so it has poles that are different. And if we were to measure the difference in a typical human cell, we would find that the number would be negative. And from here on out, we're going to focus just on neurons. So the typical resting membrane potential of a neuron is minus 70 millivolts. So again, minus 70 millivolts tells us that we have more negative charges on the inside and more positive charges on the outside. And the 70 is an actual measurement of that difference in charges. And again, think of the minus 70 as the cell being in a polarized state. So, if I were to add positive charges to the inside of the cell, I would make the inside more positive, and I would make it look more like the outside. So I would make the charges look more equal. This would make the membrane potential increase or go towards 0, because again, remember, once they equal out, you're going to have a 0 membrane potential. So adding positives to the inside of the cell would make the membrane potential become more positive, and we call that depolarization, because we are making it less polarized. We are making the charges more equal. So that is depolarization, is a more positive membrane potential. Likewise, if I were to take positive charges away from the inside of the cell, so if I were to move positives from the inside to the outside, I am making the difference even greater. Now I am making the inside even more negative compared to the outside. So I am making the difference in charges larger. So my number would become more negative. So I am taking away positive charges. So the number becomes more negative than minus 70. And I would call this hyperpolarization, meaning that I am making it even more polarized. Remember, hyper means more. So hyperpolarization, I'm making an even greater difference in the charges. So hopefully this will help you understand some of this terminology as we move forward. Okay, now let's talk about sodium's electrochemical gradient. So stick with me. If you understand this background information, it's going to make understanding all the PhysioEx concepts that much easier. So here is a picture of our typical cell, again with more negative charges on the inside and more positive charges on the outside. So as you learned earlier this semester, the concentration of sodium is high on the outside of the cell and low on the inside of the cell. Sodium is also a positively charged ion. So if we first consider sodium's chemical gradient, that is where it is in high concentration and where it is in low concentration, Because sodium is high on the outside of the cell and low on the inside of the cell, the chemical gradient favors sodium moving into the cell as it diffuses. So again, diffusion is movement from an area of high concentration to an area of low concentration. And if we just look at the charges, sodium is a positive charged ion, and positive charges are attracted to negative charges. So if we just look at charges, sodium is also attracted to the inside of the cell, where it is negatively charged. So sodium has what we call two driving forces that make it want to go into the cell. First, going into the cell means it moves from high to low concentration. And second, going into the cell means its positive charges can meet up with the negative charges on the inside. So overall, sodium has what we call a high driving force for wanting to go into the cell. What this means is that anytime you open a sodium channel in the membrane, sodium is going to want to rush into the cell. So what will this do to the membrane potential? So just a reminder, the normal resting membrane potential of a neuron is minus 70 millivolts. And remember that 0 represents an equal charge, so that there is no difference in charges between the outside and inside. And so if we open a sodium channel, and it doesn't matter if it's mechanically gated channel, ligand gated channel, or voltage gated channel, or even if it's a leak channel, anytime you open a sodium channel, sodium is going to want to rush into the cell. So we will be adding positive charges to the inside of the cell. That will make the membrane potential more positive, so it will go up. And in fact, it's helpful to remember that sodium's equilibrium point, which is the point at which the sodium molecule is perfectly happy, it's equal concentrations and it's equal with the electrical charges, its equilibrium point is all the way up at plus 66 millivolts. So that means sodium really wants to come into the cell. It wants to take that cell membrane from minus 70 millivolts all the way up to plus 66 millivolts, at which point you actually have more positive charges on the inside of the cell than on the outside. All right, so keep in mind that when you open a sodium channel, and it doesn't matter what kind, sodium rushes into the cell and causes depolarization. So that's fact number 1 to keep in your head. Now let's consider potassium. If you recall from earlier this semester, potassium has a high concentration on the inside of the cell and a low concentration on the outside of the cell. So first, let's consider potassium's chemical gradient. Again, the chemical gradient is based on the concentrations, and if we just look at concentration, potassium wants to move out of the cell. So it wants to go from an area of high concentration to an area of low concentration, and this would favor the movement of potassium to the outside of the cell. But now let's look at potassium's electrical gradient. Potassium is also positively charged, meaning that it is attracted to negative charges. So potassium's electrical gradient would favor the movement of potassium into the cell, where you have more negative charges. So unlike sodium, potassium has two opposite driving forces. One driving force favors it moving out of the cell, and the other driving force favors its movement into the cell. If we were to add these two together, overall, potassium has a small driving force for it to leave the cell. So to move from the inside to the outside of the cell. It doesn't have as strong of a driving force as sodium, so it doesn't want to move as rapidly or as quickly out of the cell as sodium wants to move into the cell. Nevertheless, if you open a potassium channel, regardless of the type, potassium will flow out of the cell. So let's think about what this will do to the potential. So again, we have our scale over here. with the resting membrane potential of minus 70. We also show the 0 millivolts, which is the no difference in charges. But in this case, if we open a potassium channel, potassium leaves the cell. So we are subtracting or taking away positive charges from the inside of the cell, making the inside of the cell more negative. So we are making it hyperpolarized, or even a larger difference between the positive and negative charges. So just like sodium, potassium also has a, quote, happy place, which is its equilibrium potential. And it's helpful to remember that potassium's equilibrium potential is minus 90 millivolts. So that means that potassium would leave the cell until the membrane potential reached minus 90 millivolts, and then it's happy and at equilibrium. So you'll notice that potassium doesn't want to move as far away from the resting potential as sodium. This is why we say that it has a smaller driving force. But the key take home to remember here is that if you open any type of potassium channel, it could be a leak channel, or it could be an open mechanically gated channel, ligand gated channel, or voltage gated channel, potassium will move out of the cell. The membrane potential will become more negative or hyperpolarized. Let's recall a few numbers that were covered in the background information section. Recall that at 0 millivolts, the cell is not polarized. It has equal charge concentrations. The resting membrane potential is minus 70 millivolts, which means that the cell is more negative on the inside and positive on the outside. Sodium has an equilibrium potential of positive 66 millivolts, meaning that if you open a sodium channel, sodium will want to rush into the cell, making the inside more positive until it goes all the way up to plus 66. And potassium's equilibrium potential is negative 90 millivolts, meaning that if you open a potassium channel, potassium will want to move out of the cell, making the inside of the cell more negative down to negative 90. So, the resting membrane potential is what it is because the membrane at rest has leak channels in it. And recall that leak channels are channels that are always open, and so ions are always able to move through these channels. So at rest, sodium is able to leak into the cell, and potassium is able to leak out of the cell. The fact that the resting membrane potential is negative 70 millivolts, and it is much closer to potassium's equilibrium potential, tells you that there are far more potassium leak channels than there are sodium leak channels. So that is why the resting potential is close to potassium's happy place or equilibrium potential. It's not exactly the same because there are some sodium leak channels that are making the membrane more positive, which brings it up to a negative 70. There is one more final point to cover with the resting membrane potential before we move on. But Dr. Sawyer, you might say, if the membrane has leak channels and sodium is always leaking in and potassium is always leaking out, then how can we maintain these concentration differences to allow the neuron to work? Well, great question. This is where the sodium-potassium pump becomes very important. We talked about this pump earlier in the semester, and if you recall, the sodium-potassium pump uses 1 ATP to pump 3 sodiums out of the cell and to pump 2 potassiums into the cell. So even though these ions are constantly leaking, the sodium-potassium pump is putting them back to maintain high potassium inside the cell and high sodium outside the cell. If these pumps ever stopped working, your neurons could not function and you would die. So the resting membrane potential is dependent on 2 things. The first is that you have leak channels, and you have far more potassium leak channels than sodium leak channels. And the second thing is that you have the sodium-potassium pump, which is correcting the leaks by putting the ions back in their original places. What I really want to focus on for this lesson is the principle of graded potentials. So keep in mind that graded potentials are produced by mechanically gated ion channels and ligand-gated ion channels. But for these examples I'm about to go over, I'm going to use ligand-gated ion channels as the example. So the principle of a graded potential is that the intensity of the stimulus will determine the degree of change in the membrane potential. So let's take a look at one example here. So we have partial cell membrane showing, and I am showing 5 different ligand-gated sodium channels, and I'm also showing the sodium ions, which remember are high on the outside and low on the inside. So the idea of a graded potential is that if you have a small stimulus, in this example, let's say we have one little chemical that is present, this chemical is only going to be able to open one of these ligand-gated channels, and only a small amount of sodium is going to be able to come into the cell. Also recall from earlier lessons that when sodium comes into the cell, this causes the cell to become more positive or to depolarize. So I am showing on the little graph here, we're starting at the resting membrane potential of minus 70 millivolts, and when I have the small stimulus, I get a small depolarization, so a slight change, maybe up to negative 65 millivolts. The second example here is going to show a medium stimulus. So in this case, I have more of the chemical molecule that is providing the stimulus. So in this case, we've got 3 chemical molecules, and we're going to open up 3 of these ligand-gated channels. More channels open means more sodium can rush into the cell. The more sodium that rushes into the cell, the bigger the depolarization that you will get, or shift toward a positive potential. So we are showing here that now we are going up to, say, a negative 50 millivolts. So we got a bigger change with a larger stimulus. And then for our final example, this will be the highest stimulus level you tested. So now we have a bunch of odorant molecules or chemicals floating around. So they are going to open up all of the ligand-gated sodium channels. So that means a whole lot of sodium can come into the cell, which means I'm going to get a whole lot more positive charges inside the cell and a larger depolarization. So as we can see here, now we're going to go all the way up to minus 35 millivolts. So these changes in the resting membrane potential are called graded potentials. And the term graded potential represents the fact that the size of the change depends on the size of the stimulus. A small stimulus would give you a small change, and a large stimulus would give you a large change. There is one more key point to learn about graded potentials. So we've covered that a graded potential means that the size of the change of the membrane potential depends on the size of the stimulus. But the other key point is that graded potentials can be either depolarizing or hyperpolarizing. So let's look at one more example. In this example, I'm not going to use the animations. I'm just going to show the after pictures. So let's start with this picture here, showing a cell, and now we are just showing the potassium. So remember that potassium is of a higher concentration on the inside, lower concentration on the outside. And these are ligand-gated potassium channels. And the little green molecule is our stimulant, so our chemical stimulus. So in this particular image, we see that there is a small stimulus, meaning that only one chemical molecule is around to bind to one of the ligand-gated potassium channels. So only a little bit of potassium is able to leave the cell. And remember that as potassium leaves the cell, this causes the inside of the cell to become more negative. And so that is a hyperpolarization. So again, if we show our little scale, and we show that we start at a minus 70 millivolts, which is our resting potential. If we make the inside of the cell more negative, now we are going to have a downward change, so a change toward the more negative area, and this is called a hyperpolarization. So in this case, a small stimulus that activated ligand-gated potassium channels will cause a small hyperpolarization. So here is our second example. In this example, we have a medium stimulus as shown by 3 of the chemical ligands. And so now we are opening up 3 of the ligand-gated potassium channels. So more potassium is able to leave the cell, making the inside more negative. So if we show that on our scale, this would look like a medium hyperpolarization. So now we're going from minus 70 to say minus 80. And then in our third example, this would represent a large stimulus. So we have a bunch more of the chemical stimulant molecules. Now all of the ligand-gated potassium channels are open. So a lot more potassium is able to leave the cell, which makes the inside even more negative. And so this would look like a large hyperpolarization. In this example, we're going all the way down to negative 90 millivolts from negative 70. So the key here is that graded potentials are changes in the resting membrane potential that depend on the size of the stimulus. And the change can either be in the positive direction, which is a depolarization, or it can be in the negative direction, which is a hyperpolarization. So opening sodium channels, it could be a mechanically gated sodium channel or a ligand-gated sodium channel. Opening sodium channels will cause graded depolarizing potentials. And opening potassium channels, it could be a mechanically gated potassium channel or a ligand-gated potassium channel, but opening potassium channels cause graded hyperpolarizing potentials. Which type of channel is opened depends on the neuron and the specific situation, and that is beyond the scope of the lessons that we're going to learn in this class. Your PhysioEx exercise doesn't really introduce voltage-gated sodium channels until activity 4, but I feel that it is necessary and better to help you understand this material to go over here. So first, a voltage-gated sodium channel can be abbreviated VGSC, and I am okay with you using abbreviations on the practical as long as you get them 100% correct. But I know writing out voltage-gated sodium channel is a lot to write out. So these channels are gated. That means they have gates, and we'll take a look at a picture in a moment. And they open and close based on changes to the membrane potential. And voltage-gated sodium channels have 3 states, which is going to be important for understanding in a little bit not only how the action potential works, but how the refractory periods work. So let's take a look at a picture of a closed voltage-gated sodium channel. So in this condition, no sodium ions can pass through this channel. Voltage-gated sodium channels close at around resting membrane potential, which remember is minus 70 millivolts. From the closed position, voltage-gated sodium channels can then open. They open at what we call the threshold voltage, which is typically slightly more depolarized than resting. So the typical threshold voltage is around minus 60 millivolts or minus 55 millivolts. It can depend on the neuron, and we'll talk more about threshold voltage when we actually get to this activities experiment. From the open condition, voltage-gated sodium channels actually have a second gate, and so the next state that they go to is called an inactivated state. And they inactivate at a membrane potential of positive 30 millivolts. So this will become important when we discuss the action potential in the next section. And then from the inactivated state, they will go back to the closed state. Again, this will happen at resting potential of minus 70 millivolts. So the sodium channel has to go through these three states in this order. It has to go from closed to open, from open to inactivated, and then inactivated to closed. It cannot skip steps. So for example, it can't go from open straight to closed. The voltage-gated potassium channel I'm going to go ahead and introduce here as well. It's a little simpler, but because it is a voltage-gated channel, that means that it does open and close, again based on changes to the membrane potential. And the voltage-gated potassium channel has two states. So here is a picture of the voltage-gated potassium channel when it's open. And when it's open, it'll allow potassium to leave the cell. And note that the voltage-gated potassium channel opens when the membrane potential reaches positive 30 millivolts. That's what is required for it to open. And then it goes straight from open to closed, and it closes at around resting membrane potential, so at around minus 70 millivolts, but it closes kind of slow. And again, that'll become important when we take a look at the action potential. And so this channel goes straight from closed to open and then open to closed, so it only has two states. Okay, now you have enough information, if you've done all of the sections up until this one, to really understand the action potential and what is happening. So I have our little graph set up here, and I have some important membrane potentials on the y-axis. So we're going to start out with the negative 70 millivolts. So remember, that is the resting membrane potential of a neuron. So I'm going to draw a little line here to show that my neuron is hanging out, not doing anything, and he's at minus 70 millivolts. So that is the resting potential. Now the negative 55 millivolts that I have over here is the threshold potential for this particular neuron. So on these types of graphs, threshold potential is usually shown by a dotted line. So I'm going to put a dotted line here, and again, remember that this is representing the threshold potential. Now, as we talked about in the previous activity, you cannot get an action potential until you reach threshold voltage. And here we are hanging out, and we're at minus 70. So how does a neuron get from minus 70 millivolts to the threshold of minus 55 millivolts? Yes, we need some type of a stimulus and a graded potential. More specifically, a graded depolarizing potential. So we need to open up either some ligand-gated sodium channels or some mechanically gated sodium channels. This will allow enough sodium to come into the neuron to raise the membrane potential up to threshold. So I am putting this little line on the graph. This represents the graded depolarizing potential. So again, remember that this is caused by either ligand-gated sodium channels or mechanically gated sodium channels. Now I am at the threshold value of negative 55 millivolts. So at this point right here is when the voltage-gated sodium channels open. So remember, these channels will only open at threshold voltage. Now, once they are open, a lot of them open, and you have a whole bunch of sodium that comes rushing into the neuron. And as the sodium rushes into the neuron, it causes more voltage-gated sodium channels to open nearby. And so soon you have a whole bunch of channels open and a whole bunch of sodium rushing in. And this is what we call the depolarization phase of the action potential, which is represented here on the graph. So the key here is that during the depolarization phase, the voltage-gated sodium channels are open, and you have active depolarization of the membrane, meaning that the membrane potential is becoming more positive. Now notice that we went up to plus 30. Now in a previous lesson, I explained to you that sodium's happy place, or its equilibrium potential, was at plus 66 millivolts. So sodium would be happy to keep coming into the cell all the way up to plus 66. But something happens at plus 30 millivolts. If you recall from the previous lesson, plus 30 millivolts is the trigger for the voltage-gated sodium channels to inactivate. So at plus 30 millivolts, sodium can no longer come into the cell. And there's something else that happens at plus 30. Again, if you recall the previous lesson, voltage-gated potassium channels open at plus 30 millivolts. So at this point on the graph, the sodium channels are inactivated and sodium can no longer come in. But now I am opening potassium channels, and yep, potassium can leave the cell. So what happens next is, remember, potassium wants to be all the way down at negative 90 millivolts. That's its happy place. So now potassium is going to rush out of the cell, and we call this the repolarization phase of the action potential, because we are going back to the polarized state of minus 70. So during this phase, the voltage-gated potassium channels are open, and potassium is leaving the cell. Now at this point down here, we are back at the resting membrane potential. So at this point, the voltage-gated sodium channels close. Remember, they have three phases: open and activation and then closing. So here they close, and the voltage-gated potassium channels also start to close, but they are like swinging doors at a mall that close really slowly. And as long as they're a little bit open, potassium is still going to rush out of the cell. So as the potassium still leaves the cell, the membrane still becomes more and more negative. And that leads to this phase here, which is the hyperpolarization phase. So because the voltage-gated potassium channels are slow to close, potassium can still leave the cell. And so that's going to make the membrane potential more negative, and that's going to lead to this little dip here. Now eventually those channels will close. The potassium can no longer leave the cell except through leak channels, and then your leak channels and your sodium-potassium pump is going to put the cell back to the resting membrane potential of negative 70 millivolts. So there you have it. That is the action potential. This activity dealt with the refractory periods of the action potential. So the refractory period is a period in which it is more difficult or impossible for a particular cell to fire a second action potential. So let's first talk about the absolute refractory period. The absolute refractory period is the period after an action potential when a membrane of a cell cannot generate a second action potential to any type of stimulus. It doesn't matter if it's a small, medium, or large stimulus, that membrane of that cell is not going to have a second action potential during this period. And it occurs from the point when the voltage-gated sodium channels open until the point when they close, as shown here on the graph. So remember, this pulls together a couple of concepts that we've already looked at. One concept is that you cannot get an action potential unless you can open voltage-gated sodium channels. During this period that is highlighted in the graph, the voltage-gated sodium channels are either already open or they are inactivated. And so when they are inactivated, remember, they cannot open again. They have to go through that three-step process. They have to go from open to inactivated to closed before they can open again. So if they are already open or they are inactivated, they cannot open again, and thus you cannot get another action potential. So during this period, it is impossible to get a second action potential, because think of the voltage-gated sodium channels as being out of commission during this time frame. Now let's look at the relative refractory period. This is the period after an action potential when the membrane of the neuron can only generate a second action potential if the stimulus is larger than a normal stimulus. So I'm going to highlight this region on the graph, and then I'll explain why this is. So first, notice that the region of the relative refractory period occurs while the membrane is hyperpolarized. That is, the membrane potential is more negative than resting. So again, this helps us to review a couple of concepts. Remember that under normal conditions, to get an action potential, I have to have a depolarizing graded potential that raises my membrane potential up to the threshold value. So in this example graph that I'm showing here, I would need a 15 millivolt stimulus that would allow my membrane potential to go from minus 70 to minus 55 under normal conditions. When the membrane is hyperpolarized, as during the relative refractory period, I am farther away from threshold. So at this trough down here, at this little valley, I'm at negative 80 millivolts. I still have to get to negative 55 in order to open voltage-gated sodium channels, but now I need a stimulus that is 25 millivolts in order to let me make that large of a change so that I can hit threshold. So that is why we call this the relative refractory period. I can get a second action potential, but the stimulus has to be larger than normal. So over here I would have only needed a 15 millivolt stimulus, but at this point I would need a 25 millivolt stimulus. So that is the relative refractory period. So you should have a pretty good understanding now of how all of the neurons in your nervous system communicate. The principles that we went over in this PhysioEx are just repeated over and over again from one neuron to another. So you start with all of the neurons at a resting membrane potential. Then in the dendrites and the soma, you have graded potentials, which are driven by the ligand-gated channels and the mechanically gated channels. These will generate action potentials that will travel along the axon. Then when you get to the synaptic terminal, this is where you'll have the synaptic transmission. The release of the neurotransmitter will cause a graded potential in the dendrites or soma of the next neuron, which again may generate action potentials that will travel along its axon, which again will get to the synaptic terminals and called neurotransmitter release, which again will cause a graded potential in the dendrites and soma of the next neuron, and on and on and on. So you can now better understand how the billions of neurons in your nervous system communicate with each other. And one last little thing to think about. You know that my background is neuroscience, and so I find all of this extremely fascinating. And even if you found this lesson somewhat confusing, if you hated it, if you loved it, if you were fascinated by it like I was, any of the emotions or thoughts that you had about this lesson would not be possible without action potentials, graded potentials, and synaptic transmission going on in your brain at this very second.